settle in and let your breathing slow, because the lab is finally quiet, the kind of quiet that hums a little from an old fluorescent light that refuses to retire. In front of you, a microscope waits, patient, unjudging, like a small tunnel into a world that never learned how to be tidy. You think soil is just dirt, plain, forgettable. But under glass, it becomes a careful biography. Mineral crumbs, soft plant ghosts, and tiny hitchhikers that traveled on a shoe without permission. Tonight, with the Sleepless Scientist keeping the pace gentle, you will watch ordinary grains turn into calm, useful clues and let your thoughts settle the way dust always does. You are here after hours, when the building has stopped pretending it is alive. The heavy doors have already sighed shut. The hallway lights are dimmed, and in this room, the most dramatic thing happening is that the fluorescent tube keeps buzzing, like it wants to be included. On the bench in front of you, there is a small lineup of evidence containers, not the glamorous kind. No glowing vials, no high-stakes movie nonsense. Just little paper packets, a few plastic tubes, and a slide box that clicks softly when you open it. This is where forensic soil begins, with something boring enough to ignore. And that is why it works. Because soil is not one substance. It is a mixture that remembers where it has been. It remembers the rock that broke down into it. It remembers the plants that dropped into it. It remembers the water that moved through it, and the air that dried it, and the boots that mashed it into a path. In an investigation, soil is often the quiet witness. It cannot talk. It cannot lie. It can only cling and smear and hide in the seam of a shoe, waiting for someone patient enough to look. So you start with routine. Routine is how you keep your mind calm and your work honest. You label first. Case number, sample number, date, location if you have it. Your handwriting is slower than usual, not because you are dramatic, but because mistakes happen when you rush. Soil evidence is often tiny. Sometimes you get a pinch of dust from a car mat, and that is all you have. Then you clean, not obsessively, just carefully. A slide gets wiped. A cover slip gets checked for smudges. Tweezers get a quick pass with alcohol. It is all very unromantic, and that is exactly what keeps it real. You cannot interpret a world that you have contaminated. Now you take the first sample packet and open it like it might sneeze. A faint, dry smell rises. Not a strong smell, just a suggestion. If you have ever opened a bag of potting soil or scuffed your shoe on a dusty sidewalk, you already know it. It is the smell of minerals and old leaves and time. You tip a little onto weighing paper. Under the lamp, it looks like dirt, brownish, a few darker specks, a few pale grains that catch light. Nothing that would make anyone gasp. Congratulations. You have discovered the entire reason microscopy exists. With the naked eye, soil is a blur. Under magnification, it becomes a crowd. And forensic work likes crowds because crowds contain differences. A single grain can be generic. A mixture of grains can be oddly specific. You take a small amount, the smallest amount you can manage without feeling ridiculous, and you place it on a slide. A drop of mounting medium if you are preparing something more permanent, or just a dry mount if you are doing a quick look. Then the cover slip goes down, slow, like setting a tiny window over a sleeping landscape. Your fingers know the motion. You have done it so many times that it feels like a ritual. Slide, sample, cover, press gently. Do not grind. Do not crush. Because even the act of preparing the slide can change what you see. Now, before you even look through the microscope, you do something that seems almost too simple to matter. You look at the soil in the packet again, under normal light. You note its color, but you do it honestly. Not poetic, not earthy. You note it like someone who has learned that language can drift. Is it more reddish-brown, like iron staining? Is it more gray, like ash? Or clay? Is it nearly black, like rich organic material? Or soot? Does it have visible sand grains, glittering like tiny sugar crystals? Or does it look dull and powdery? If you are lucky, you may even notice a few obvious intruders. A green fragment that could be plant matter, a shiny thread that could be fiber, a pale flake that could be paint. Those intruders are not always meaningful, but sometimes they are. And the boring truth of forensic soil is that it rarely solves a case alone. It does not kick down doors. It does not deliver dramatic monologues. It just adds weight. It supports. It disagrees. It says, quietly, this seems likely, or this seems off. Now you finally lean in. Your forehead settles against the microscope rest. Your eye finds the eyepiece. The world narrows to a circle of light. At first, it is out of focus, a soft blur of beige and shadow. Then you turn the focus knob slowly, and the blur becomes edges, and you see the first truth of dirt. Nothing is smooth. A grain that looked like a plain dot with your bare eye is now a jagged little shard. Another grain is rounded, like it has been rolled around by water for a very long time. A dark fragment has a torn edge, like burnt wood. A clear grain flashes as you tilt the stage like a tiny piece of broken glass. You thought you were looking at dirt. Wrong. You are looking at a broken world, sorted and resorted by physics. And you do it gently, because under this lens, your hands feel huge. Every movement of the stage is a slow earthquake. Every nudge of the slide is a landslide. The microscope does not care. It will show you what is there, whether you are ready or not. So, you start with the most basic setting, bright field illumination, plain, honest light passing through or reflecting off grains. You keep the magnification modest at first. You are not hunting for drama. You are surveying a neighborhood, and the neighborhood has three main personalities, even when they blend together: sand, silt, and clay. Those words sound simple, like something you learned once and forgot. But they are not just categories. They are behavior. Sand is the big one, the obvious one. Under the microscope, sand grains can look like clear crystals, milky chips, or colored fragments. Many are quartz, because quartz is annoyingly durable. Quartz survives what other minerals cannot. It is the coworker who never quits. Quartz grains can be angular, like they were freshly broken from rock, or they can be rounded, like they have been tumbled in a river, or polished by wind. Those shapes matter, not as magic clues, but as hints. Angular suggests less transport, less time being knocked around. Rounded suggests travel, maybe water, maybe wind, maybe long-distance patience. Silt is finer. In your imagination, it is like flour, and that is not a terrible comparison. Silt grains are small enough to stay suspended in water longer than sand. They settle slowly. They drift. Under magnification, silt can still show mineral edges, but the scene becomes busier, more crowded, more like a city skyline made of crumbs. Clay is the smallest, and it behaves like something half physical, half emotional. Clay particles are so tiny that you do not always see them as individual grains in ordinary light microscopy. They clump, they smear, they coat other grains, they make everything stick. Clay is not just small sand. Clay is chemistry. Clay minerals have layered structures, and they carry electrical charges along their surfaces. That means water clings to them. Ions cling to them. Nutrients cling. Toxins cling. If sand is a pile of marbles, clay is a stack of damp playing cards. In soil, clay is the secret binder. It is why some soils feel slick when wet and crack when dry. It is why some ground holds water like a sponge, and other ground drains like a sieve. And in forensics, texture matters because it changes what soil does when it travels. Sandy soil tends to fall away. It pours out. It does not cling as well. Clay-rich soil clings like it was designed to be annoying. It lodges in tread patterns. It sticks under fingernails. It stays on fabric. Congratulations! The dirt has become a personality test. Now you shift the slide slightly and you see something darker, a fragment that does not look like mineral. It has a softer edge. It looks fibrous. This is organic matter. Bits of plant tissue, tiny pieces of leaf, maybe a fragment of root. Sometimes it is so decomposed it looks like a dark smear, a soft ghost of biology. Soil is not just ground rock. It is also dead life, shredded and recycled. The scientific word is humus, spelled like humus. And it is not the tasty kind. It is the result of microbes and fungi and time breaking things down until the original shape is mostly gone. Under a microscope, humus can look like irregular dark flakes or brown clumps or thin threads. It is messy. It is the opposite of a clean mineral grain. And that mess is information. A soil rich in organic material often comes from a place with a lot of plant growth and slower decomposition and the right moisture. A soil with very little organic matter might come from a dry area or a heavily disturbed area or a place where plants struggle. None of this is certain. Soil does not hand you a labeled map. It only gives you probabilities, and you build them carefully. Now you adjust the light. You lower the condenser slightly. You change the angle. The grains pop a little more. Tiny sparkles appear. Some minerals reflect light differently. Some are translucent. Some are opaque. And you notice that even two soils that look the same to the naked eye can have different fingerprints. Under glass, one sample might be full of shiny, reflective grains, mica, little plates that glint like tiny mirrors. Another might have more dark, heavy minerals like magnetite or hornblende, making the field look peppered. Another might show pale fragments that look like tiny shells if the soil formed in a marine environment or contains crushed limestone. So when someone asks, "Is this soil from that place?" the most honest answer is not yes or no. The honest answer is, how similar is the mixture and how common is that mixture? Forensic soil is rarely about uniqueness. It is about consistency. Now you pull back for a moment and consider the quiet comedy of it. A person can spend their whole day trying to be a clean, modern human, and the ground still collects them. A shoe steps into a patch of soil, and it becomes a sampling tool. Not on purpose, it just happens. Tread patterns trap grains. Mud dries in layers. Tiny particles wedge into seams and stitching. If you want to understand soil transfer, you can imagine it as a kind of accidental shopping trip. You walk through a landscape, and the landscape sells you small souvenirs you never asked for. And those souvenirs matter most when the question is simple. Could this person have been there? Could this vehicle have been here? Could this object have touched the ground at that location? The science is not glamorous. It is careful comparison. It is not a single perfect match. It is a set of small agreements that stack up until the story becomes hard to ignore. Now you take a second sample just to see how quickly dirt stops being generic. This one is lighter in color, more tan than brown. You mount it, you focus, and immediately you see more sand-sized grains, more clear quartz, and fewer dark organic flakes. You also see something you did not see before. Little rounded grains that look like tiny beads, perhaps ooids, or maybe just well-rounded sand. Or you might see a grain with a pitted surface, like it was etched. Sometimes those surface textures tell you about transport. Wind-blown sand can show frosted surfaces and tiny impact marks, like it has been sandblasted by the atmosphere. River-transported grains can be smoother and more polished. Even without going deep into specialized techniques, your eyes can learn to notice the difference between freshly broken and patiently worn. And this is where the quiet lab routines become comforting. You are not rushing. You are not chasing a plot twist. You are just noticing. You write down plain notes. More clear grains, fewer dark fragments, grains mostly rounded, some shiny plates. You would be surprised how useful boring notes can be. In a world full of dramatic storytelling, a sentence like "more shiny grains than sample A" can be the difference between clarity and confusion. Now, because this is microscopy, you have another lever you can pull: light. You switch gently to polarized light. If your microscope has polarizing filters, you bring them into place. The field changes. Minerals that were plain suddenly become lively. Some grains glow with interference colors, pale yellows, soft blues, sometimes a strange pink. They shift as you rotate the stage. The colors are not decoration. They come from birefringence, a property of some crystals, where light travels through them at different speeds depending on direction. Translated into human language, the crystal is bending light in a way that reveals its internal structure. Quartz will often show low-order gray and white under crossed polars. Feldspar can show different colors and sometimes twinning faint stripes or patterns inside the grain. Mica can light up strongly depending on thickness and orientation. It is quietly beautiful, like stained glass that forgot it was supposed to be in a cathedral. But the beauty is not the point. The point is identification. Under polarized light, your ability to tell minerals apart improves. You still need experience. You still need reference materials. You still need restraint. But you gain another layer of information. And in forensics, layers are everything. Now, because you are not here to get overstimulated, you keep it simple. You watch a few grains change color as you rotate the stage. You notice how some stayed dark no matter what. Those may be opaque minerals or simply grains oriented in a way that does not transmit light. You make a note and then you go back to bright field because your eyes and brain deserve consistency. You are building a mental library, slowly. And speaking of libraries, soil is one. Soil is a library made of dust, but the books are not bound. They are shredded, they are mixed together, they have been rewritten by worms and roots and rain. Every place has a slightly different collection. Even two spots in the same yard can differ if one spot gets more shade, or more foot traffic, or more runoff. Soil is local and personal in a way that sounds sentimental, until you remember it is also indifferent. It does not care who you are. It clings anyway. Now, imagine a shoe print at a scene, not the dramatic kind, not a perfect soul impression in cinematic mud, just a scuffed mark or a dusty footprint on a floor. The soil in that tread is a sample. It might contain grains from the immediate area. It might contain grains from somewhere else. If the shoe has traveled, it might be layered with older soil packed deep and newer soil on top. So one of the first questions a careful examiner might ask is not, what is this soil, but how many soils are here? Sometimes you can see it directly. A darker, clay-rich smear underneath, and a lighter, sandy dust on top. Sometimes you need to separate it gently. Sometimes you cannot separate it at all. Congratulations, your evidence is a smoothie. Now you zoom in again and look for the subtle extras. The things that are not just minerals. A tiny plant fragment can tell you something about environment. A thorny bit of leaf, a fragment of moss, a little piece of wood. Sometimes you may find diatoms in a soil from near water. Diatoms are microscopic algae with silica shells like tiny glass boats. If you find them, it can suggest a wet environment or at least contact with water. Sometimes you may find charcoal fragments, black and porous, suggesting fire exposure, or human activity, or simply a natural burn in the area. Sometimes you find small, bright fibers, cotton, wool, synthetic. Under a microscope, fibers have their own textures. Cotton can look like a flattened ribbon with twists. Wool can have a scaly surface. Synthetics can be smooth and uniform, sometimes too perfect. A single fiber does not convict anyone, but it can suggest contact. It can suggest transfer. It can suggest a shared environment, like a car interior, a workshop, or a home. And sometimes, yes, you find glitter. Glitter is the herpes of craft materials. It spreads, it persists, it does not forgive. If you find a tiny reflective flake in soil, it could be mica, or it could be paint, or it could be glitter from a clothing item, or cosmetic. You do not guess. You note it, you compare it, you treat it like any other particle. Forensic work is not about being impressed. It is about being consistent. Now you think about how soil gets from place to place, and it is not always dramatic. Wind is a quiet thief. It lifts fine particles, especially silt and clay, and carries them. Over time, wind can sort grains like a patient librarian. In deserts, dunes form where wind drops sand it cannot carry further. The sand in dunes is often well sorted. meaning the grains are similar in size, because wind is picky. It does not like carrying mixed luggage. Water is less picky. Water can carry a wide range of particle sizes depending on how fast it moves. A calm stream may carry mostly fine material. A flood can carry gravel. Water rounds grains by tumbling them. It also mixes layers, especially during storms. Then there is the most chaotic transport mechanism of all. Human movement moves soil constantly. On shoes, on tires, on pet paws, on clothing cuffs, in pockets, in the grooves of a phone case, in the tiny textures of a backpack strap. You do not notice it because it is small and because modern life trains you to ignore dirt. But it is happening, quietly, always. That is why forensic soil work often begins with very gentle questions. Could this soil have come from that place? Is this consistent with the soil near the scene? Is there anything unusual here that suggests a specific environment? Unusual does not mean rare. It just means distinctive in a helpful way. Now you tilt your head and let your eyes rest for a moment. Looking into a microscope is a kind of tunnel vision, literally. It is easy to get lost in the circle of light. So you break the trance the way experienced people do, by zooming out. You look at the entire slide again at low magnification. You scan across. You note the distribution. Are the grains all similar in size, suggesting sorting? Or are they mixed, suggesting disturbance? Are there clusters of dark organic matter? Are there patches where fine material coats larger grains, suggesting clay content? These patterns are not just pretty. They are clues about how the soil formed and how it moved. Now, because this is forensic work, you also think about the chain of custody, not as a dramatic courtroom concept, but as a simple reality. If you mix samples, you ruin them. So you keep tools separated. You clean between samples, you close containers promptly, you work on one sample at a time, you label everything. And when you feel bored, you remind yourself that boredom is protective. Bored people follow procedure. Excited people skip steps. And skipping steps is how you accidentally create evidence that never existed. Now you return to the idea of soil as a personal history. Not personal like it has feelings, personal like it is specific to a place. A soil from near a construction site might contain cement dust, small, pale, angular fragments. A soil from near a road might contain brake dust, tiny dark metallic particles, and bits of asphalt. A soil from a garden might contain fertilizer granules, perlite, or bits of mulch. Even urban soil has layers of human influence. Sometimes it is almost entirely human-made fill, crushed brick, glass, concrete, and compacted dust. Under a microscope, it looks like a scrapyard in miniature. And if you are working an investigation, those human-made particles can be extremely useful. Not because they are rare, but because they narrow possibilities. If a soil has lots of brick dust and mortar fragments, it suggests older urban environments, demolition areas, or specific types of construction. If a soil has lots of rounded quartz and very little organic matter, it might suggest a beach or dune environment. If a soil has a rich, dark organic content and lots of plant fragments, it might suggest forest floor or well-developed topsoil. Again, none of this is a magic stamp. It is a gentle accumulation of hints. Now, speaking of hints stuck to you, the microscope is where those tiny clues stop being dirt and start being details. So you lean in again and you decide to meet the microscope properly, the way it likes to be met, slowly. You adjust the coarse focus until the grains are near clarity. Then you switch to fine focus. The knob turns smoothly and the image breathes. Blur, sharp, blur, sharp. It is a little hypnotic if you let it be. And this is where you notice something that always surprises people the first time. Under magnification, a soil grain can look like a landscape. A crack becomes a canyon. A flat surface becomes a frozen lake. A jagged edge becomes a cliff face. A tiny bubble in mounting medium becomes a pale moon drifting across the field. Your brain wants to make stories out of it. That is normal. Brains are desperate narrators, but you keep one part of your mind practical. You ask, gently, what you are really seeing. Is that grain translucent, suggesting quartz? Does it have cleavage planes, suggesting feldspar? Is it flaky and reflective, suggesting mica? Is it opaque and heavy-looking, suggesting a dark mineral? If you have a little training, you can learn to recognize common mineral types. If you do not, you can still do something very useful. You can compare. Because forensics is not always about naming every grain. It is often about noticing that two samples have similar mixtures. Now you move the slide, and you see a grain that looks almost metallic, dark, opaque, with a strong reflective surface. If you had more tools, you might test for magnetism, because magnetite is magnetic. A tiny magnet can sometimes pull magnetic grains out of soil, like a quiet trick. But tonight, in this calm lab, you are just observing, not doing stunts. You make a note, opaque, dark grains present, possibly heavy minerals. Then you keep scanning, and you see something like a tiny sphere, perfectly round. That can happen naturally, but perfect spheres in soil often suggest industrial sources, like fly ash from combustion. Fly ash particles can be spherical because they formed as molten droplets that cooled in air. That is one of those details that can connect soil to human activity. A soil near a power plant or near heavy industry can contain more of these particles. A soil from a remote forest might not. Again, not proof, just a pattern. Now your mind drifts briefly to the idea that a single step collects all of this. Not just minerals, but human history. The dust of roads, the powder of construction, the ash of combustion. You are a walking sampler of the modern world, and you never asked to be. Congratulations. You are evidence. Now you remind yourself that forensic soil work is careful and humble. You do not overstate. You do not claim certainty where there is none. Because soil is messy, literally and statistically. Even if two samples share many features, they may still come from different places because many places share similar geology and similar human disturbances. And the reverse is also true. Two samples from the same place can look different if they were collected from different layers or different microenvironments or different times after weather events. A rainstorm can wash away fine particles. Sun can bake and harden clay. Foot traffic can grind and mix layers. A lawnmower can fling soil. A dog can redecorate an entire yard in a few minutes. So, when you compare soils, you are comparing living mixtures, not static objects. But the microscope still helps because it slows the world down enough to see what the eye misses. And as your eyes adjust to this small universe, you begin to recognize the most common characters in the scene: sand, silt, and clay, behaving like they always do, even when no one is watching. Sand sits there with its sharp edges and stubborn clarity. Silt drifts into corners, fine and patient. Clay coats everything, clumping and binding like it has a grudge. You can almost predict how a soil will feel just by looking. A sand-rich soil will feel gritty, like tiny grains rolling under your fingertips. A silt-rich soil will feel smooth, like powder. A clay-rich soil will feel sticky when wet and hard when dry. And those textures matter in the real world because they control water. A sandy soil drains quickly. Water runs through it, leaving little behind. Plants may struggle unless water is frequent. A clay-rich soil holds water tightly. It can stay wet longer, and it can swell and shrink. It can create poor drainage and slow oxygen movement, which affects roots and microbes. A silt-rich soil can be fertile but prone to erosion. Fine particles can be carried away easily by water and wind. So when you see the mix under the microscope, you are also seeing how that ground behaves under rain and footsteps. You are seeing the physical rules that shape a landscape. And in forensic contexts, behavior matters. A soil that clings might transfer differently. A soil that pours might fall away. A soil that forms hard clods might break into chunks in a tread. Now you pause on a patch of the slide where grains seem coated in a fine film. That film could be clay. It could be a mix of both. Coatings matter because they can carry color. A soil can look red because iron oxides coat grains. It can look dark because organic material coats grains. It can look pale because carbonate dust covers everything. Under the microscope, coatings show up as halos and stains. They soften edges. They make grains look less clean. Clean grains often suggest active transport and washing, like in streams or beaches. Dirty coated grains often suggest soils that have been sitting, developing, interacting with water and biology. Time leaves films. Now, you let yourself enjoy the gentle repetition of focus and scanning. It is not thrilling. It is not supposed to be. It is a quiet practice in attention. And attention, applied long enough, becomes knowledge. You keep noticing the same kinds of things. A clear quartz grain with a chipped edge, a pale feldspar fragment with a slightly cloudy look, a shiny mica plate, thin and reflective, a dark organic flake with a ragged shape, a tiny fiber, smooth and bright, caught between grains like a thread in a crack. Each one is small. None is dramatic. Together, they become a profile. Now, before you go any deeper, you do one more practical thing. You remind yourself what you are trying to answer. In forensic soil work, the question is rarely, what is the exact origin point? It is usually, is this consistent with that location, or is it inconsistent? Consistency is a quieter concept than identity, but it is more honest. If a suspect claims they never left a paved parking lot, but the soil on their shoe contains clay-rich forest topsoil with leaf fragments and pollen from a plant that only blooms in early spring, that is interesting. Not because it is magical, because it suggests a mismatch, and mismatches are often easier than matches. Now you are ready to keep going, deeper into the personality of these particles. Because once you can recognize sand, silt, and clay, you can start noticing how they were made and how they traveled without ever being asked. Some grains arrive the slow way by being bullied for millions of years. You hold a piece of granite in your mind, something that once cooled deep underground, confident and solid. Then you imagine water seeping into its tiny cracks. Night comes, the temperature drops, the water freezes, and it expands like an obnoxious tenant who insists on more space. The crack widens, a fraction at a time. Do that enough winters, and the rock gives up, not dramatically, just quietly into smaller pieces. That is physical weathering. It is not poetic. It is persistence. Then you add chemistry, because chemistry always shows up uninvited. Rainwater dissolves a little CO2 from air and soil, forming a weak carbonic acid. Weak, yes, and still capable of slowly changing minerals. Feldspars can alter into clay minerals through hydrolysis, a reaction where water helps break and rebuild the crystal structure. Translated into human, the mineral gets remodeled into something softer and smaller, something that likes to cling. And when you look at a clay-rich soil under the microscope, you are seeing the end of that long argument between rock and water. Not all grains are made this way, though. Some grains arrive dramatically, like they were delivered by disaster and stamped with urgency. You are staring at a clear quartz grain, and you notice it has sharp edges, fresh-looking, like it was snapped yesterday. That kind of angularity can suggest the grain did not travel far. Maybe it came from nearby bedrock, crushed by construction, or broken by frost, or scraped loose by a landslide. Then you drift to a grain that is rounded, smooth, almost polite. That grain has been transported. It has been rolled, bumped, and ground against other grains. It has been tumbled like laundry, except the laundry is stone and the washing machine is a river. Under magnification, transport leaves signatures you can learn to recognize. Wind transport can produce grains with frosted surfaces, tiny impact pits, and a kind of matte finish. Water transport often rounds grains more smoothly and can leave them polished. Glacial transport, when it appears, can leave scratches and a crushed look, because glaciers do not roll grains gently. They drag them under pressure, like sandpaper, with ambition. And speaking of pressure, you can feel it in the way soils sort themselves. Wind is selective. It picks up what it can carry, and it drops the rest. That is why dune sand tends to be well sorted, with grains similar in size. In your field of view, well-sorted sand looks strangely uniform, like someone curated it. Water can be selective too, but it is moodier. During calm flow, it carries fine particles farther and drops sand sooner. During floods, it carries everything it can, including gravel and debris, then drops the mess when it slows. So when you see a soil sample with a wide range of grain sizes, from chunky sand down to fine silt and clay, you begin to suspect mixing. Maybe it is from a disturbed area, a stream bank, a construction site, a path with heavy foot traffic, or maybe it is simply a natural soil profile, stirred by roots and worms and time. Yes, worms, the tiny engineers nobody thanks. Bioturbation is the word. It means living things disturb sediment and soil, mixing layers. Earthworms do it, insects do it, Burrowing mammals do it. Plant roots do it. If you want a calm image, picture a slow blender that runs for years, powered by hunger. And that mixing matters for forensics because it can blur what you wish were clear layers. You might want a tidy story. Topsoil here, subsoil there. One clean horizon of time. But soil rarely performs for your convenience. It is messy and blended. And then it gets stepped on, and then it gets rained on, and then it gets stepped on again. Congratulations, your evidence has been handled by the entire ecosystem. Now your mind slides toward the simplest kind of movement, the one that happens constantly without anyone noticing. Dry soil, powdered into dust, riding air currents through a city. It settles on windowsills. It collects on shoes. It fills the corner of a car floor mat with a thin, quiet layer. This is why the location of soil inside a shoe can matter. The outer tread picks up the newest material and sheds it easily. The deep grooves can protect older soil like a little archive. The inside of a shoe can trap soil that entered once and stayed sheltered from rain. Socks can hold fine particles too, especially if someone stepped into dirt with damp fabric. In a laboratory, that means you do not just take a single scraping and call it done. You sample carefully. You note where it came from. Outer tread, inner tread, heel, toe, inside lining, because those spots can represent different times, different places, different chapters of walking. The dramatic version of this is that shoes tell stories. The realistic version is that shoes collect crumbs, and you interpret those crumbs with cautious math. Now, if you want to see transport in action, you do something almost childlike. You sprinkle a pinch of dry soil onto a sheet of paper and gently tilt it. The larger sand grains roll quickly. The silt drifts more slowly, almost floating in place before it slides. Clay does not roll much at all. It just clings, forming tiny, stubborn clumps. That simple movement mirrors the landscape. On a windy day, sand hops along the ground in a process called saltation. The grains bounce, strike other grains, and kick them forward. Finer silt can be lifted and carried as dust, traveling far. Clay, when it is dispersed, can travel too, but it often clumps because of electrostatic forces and moisture. Electrostatic forces sound fancy, but the human translation is simple. Tiny particles can stick due to charges and thin films of water. This is why clay-rich mud can cling to a boot like it has a contract. And you can see the aftermath under the microscope. Clay coatings on grains. Aggregates, little clumps of mixed mineral and organic material. Those aggregates can break apart, or they can persist depending on moisture and disturbance. A soil that forms strong aggregates might come from a stable environment with biological activity, where roots and microbes produce sticky substances that help bind particles. That sticky substance often includes polysaccharides, long sugar-like molecules produced by microbes. A microbial biofilm can act like glue. So when you see soil clumps that hold together, you are sometimes seeing the work of bacteria quietly manufacturing adhesives, because life is basically a chemistry factory that wants to keep its house from falling apart. Now, since you are here for microscopy, you let the microscope reward you with texture. You lower the light slightly and use oblique illumination, light coming at an angle. The surface details become sharper. A grain that was just a pale shape now shows pits, scratches, and little ridges. Those textures are called microtextures. They can hint at formation and transport history. Some labs use scanning electron microscopy for deep microtexture work because electron microscopes can reveal incredibly fine detail. But even with a standard light microscope, you can still get an impression. Rounded edges versus sharp edges. Frosted look versus polished look. Coatings versus clean surfaces. You are not doing absolute identification here. You are building a gentle sense of patterns. And speaking of patterns, you start to notice that soil is rarely just geology. It is also human behavior. A soil from near a road can carry tiny fragments of asphalt, black and irregular. It can carry brake dust, which may contain metallic particles from wear. It can carry tire rubber fragments, dark and elastic looking under magnification. Sometimes, with a slightly textured surface, a soil from near a building site can contain gypsum dust from drywall, cement particles, and bits of insulation. Cement particles can look pale and angular. Under polarized light, some of these materials behave differently than natural minerals. A soil from a garden can contain perlite, those bright white porous granules used to lighten potting mixes. Perlite looks like a tiny pumice, full of holes. Vermiculite can appear as shiny, layered flakes like mica, but with its own look and feel. Bark mulch fragments can show plant cell patterns even in small pieces. In other words, your slide can become a tiny map of what humans have been doing nearby, which is comforting in a bleak sort of way. Even our mess leaves clues. Now you let your attention drift to the question investigators actually care about most of the time: transfer. How does soil get from one place onto a person or object, and how long does it stay there? The answer is awkward. It depends on moisture, texture, activity, time, and luck. Wet soil transfers easily. It sticks, it smears, it fills grooves. It makes a strong first impression. Dry soil transfers too, but it sheds faster, especially if the surface is smooth. Clothing holds soil better if it has texture, like denim, wool, or fleece. Smooth synthetic fabrics can shed more, but seams and cuffs still trap particles. Hair can hold particles well, especially if there is oil or moisture. Fingernails can trap soil, especially if someone dug into ground or handled plants. Vehicle carpets are excellent collectors, basically giant filters. This is why some of the most useful samples come from boring places. The hem of pants, the seam of a backpack, the floor mat that nobody cleans, the trunk liner, the pedal area. All of those places are quiet little soil libraries. And yes, soil can persist longer than you want it to. Dried mud can stay in a tread for weeks, protected in deep grooves. Dust can embed into fabric and remain through casual wear. Washing removes a lot, but not always everything. Tiny particles can survive because they lodge where water and soap do not reach easily, which is great for evidence and terrible for anyone who wants to feel clean. Now you move forward because there is something worse than transport: mixing. A shoe can pick up soil from one place, then pick up soil from another place, and grind them together with every step. A car tire can do the same. So when you examine a sample, you might be seeing a mixture of multiple locations. This is not rare, it is normal. So a careful approach often includes looking for internal variation. You might separate a sample by gently sieving it into size fractions. Larger particles in one group, finer particles in another. You might examine different parts of a clump, outer versus inner. Sometimes the outer layer represents the newest material, and the inner core represents older material. You are basically doing archaeology in a shoe tread, and the microscope is your trowel. Now, while you are thinking about the drama of shoe treads, it is worth remembering that soil can also transfer in a way that feels almost too subtle to matter. Dust in the air can settle on objects, including clothing and hair, without anyone stepping in anything. That kind of passive transfer is why context matters. It is why investigators compare not just suspect samples to scene samples, but also consider background. What is the soil like near the suspect home, near their workplace, along common routes? Soil is everywhere, and if you are not careful, you can convince yourself a common soil is meaningful. The enemy of good forensic interpretation is overconfidence. So you stay humble, you let the microscope show you what is there, and you let your notes stay plain. Now your mind pivots smoothly toward the tiny extras you have not fully met yet, because minerals are one story, biology is another. And biology is often more specific than rock. Plants do not grow everywhere, and they do not release the same microscopic signatures. So you start looking for pollen. You do not always see it in every soil sample. Sometimes pollen is present, but sparse. Sometimes it is crushed. Sometimes it is hidden under mineral grains. But when it appears, it can look surreal. Pollen grains are like tiny armored capsules built to survive a harsh trip from plant to plant. Their outer wall is made of sporopollenin, a material so resistant that it can survive in sediments for thousands of years. The plant invests in this wall because pollen has one job, and it must not fail. Under magnification, pollen can have spikes, pores, ridges, and intricate patterns. Some look like little sea mines. Some look like smooth eggs with tiny holes. Some look triangular. Some have air sacs, like little ears, which help them float in the wind. You are looking at a plant fingerprint, a microscopic calling card. And pollen can be seasonally informative. Some plants release pollen in spring, some in summer, some in fall. So a pollen assemblage in soil might hint at the time of year a sample was collected or transferred. Though you must be cautious, pollen can persist, and it can be mixed. Still, if you find a pollen type that is uncommon in the region or strongly associated with a specific habitat, it can become useful. For example, coastal plants may produce pollen that suggests proximity to shore. Certain trees may dominate a forested area and show up heavily in local soils. Agricultural areas may contribute pollen from crops. Urban areas might show pollen from ornamental plants, the ones humans plant for aesthetics because we cannot help ourselves. The discipline of studying pollen is palynology. It sounds like the name of a quiet wizard. It is actually a serious field with reference collections and careful identification work. A palynologist can sometimes identify pollen to family, genus, or even species, depending on the plant group. And even when you cannot identify precisely, you can still note presence and abundance. Many grains of one type, few grains of another. That pattern can be compared between samples. Now you widen your search beyond pollen, because soil holds other biological fragments too. Spores from fungi can appear, sometimes small and dark, sometimes with distinctive shapes. Fungi are everywhere in soil, breaking down organic matter, forming networks, and quietly keeping ecosystems functioning. Their spores can be transported by wind, water, and animals. In some cases, spore types can hint at habitat. You might also see fragments of plant tissue, tiny pieces of epidermis with stomata, those little pores plants use for gas exchange, bits of xylem with thickened walls, the tubes plants use to move water. Under the microscope, some of these structures have recognizable patterns. You might even see tiny insects or mite fragments, parts of exoskeleton translucent and segmented. Soil is a crowded world, and death is constant. Fragments remain. If you are near water, you might see diatoms, those silica-shelled algae that look like tiny glass ornaments. Diatoms have intricate shapes, discs and boats and needles, with patterned pores. They are often associated with aquatic environments, but they can also appear in wet soils and sediments. Finding diatoms in a shoe tread might suggest contact with water, mud near a stream, a lake edge, a marsh. Again, not proof, but a hint. Now you let that settle, and you return to something even more human. Fibers. A fiber in soil is like a whisper of clothing, upholstery, carpet, rope, or industrial material. Fibers can become embedded in soil, especially in urban environments. They can also travel with a person and become trapped in soil on shoes, transferring back and forth. Under the microscope, fibers have traits: color, thickness, surface texture, cross-section shape if you have the right setup. Cotton often appears as a flattened ribbon with twists, called convolutions. Wool tends to show a scaly surface because it is made of keratin with overlapping cuticle scales. Synthetic fibers like polyester and nylon often look smooth and uniform, with consistent diameter. Some synthetic fibers have distinctive cross-sections. Trilobal, for example, meaning 3 lobes, used for certain textiles to change how they reflect light. If you can see cross-sections, you can sometimes infer fiber type. But in many soil examinations, you do not need to fully identify fiber type to learn something. You might only need to note that one sample contains bright blue synthetic fibers, while another contains none, or that both samples share a distinctive red fiber of similar thickness and appearance. You are stacking small agreements. Now you drift toward another category of tiny extras, the ones that make soil feel like modern life condensed. Paint flecks. Paint chips can be tiny, layered, and surprisingly informative. Many paints have multiple layers: primer, base coat, top coat. Under magnification, a paint chip can show those layers like a little geological cross-section. Color, thickness, and layer sequence can be compared to known sources. If you find paint flecks in soil, it might suggest a nearby painted structure, a workshop, a road marking, construction debris. Sometimes it is just background. Sometimes it is a strong associative clue. Glass fragments can appear too, especially in urban soils. Small, clear, angular shards, sometimes with curved surfaces if they came from a broken bottle. Brick fragments can show porous texture and reddish color. Plaster fragments can be pale and powdery. And then there is the category that makes forensic scientists sigh quietly. Glitter, sequins, microplastic fragments. They are visually distinctive, reflective, sometimes brightly colored. They can transfer easily. They can persist. They can be useful if you find a rare type, but they can also be so common that they become meaningless. The world has been shedding plastic for decades. Soil holds it, water carries it, air drops it. You can find microplastics in remote locations, far from obvious sources. So you treat it like anything else. Note it. Compare it. Stay cautious. Now, as you scan across your slide, you begin to see how forensic soil work is built from small acts of restraint. You could tell dramatic stories about every grain. You could claim a grain is from a river, or a desert, or a construction site, based on a single feature. That would be exciting. It would also be irresponsible. Instead, you build a calm description, grain size distribution, mineral types present, organic content, notable inclusions like pollen or fibers, overall color and texture, degree of sorting, presence of coatings. Then you compare those descriptions between samples. That is the real work, the sleepy art of comparison. So, you take 2 slides, one from the question sample, one from a known location, and you place them side by side on the bench. The microscope becomes a shuttle between worlds. You look at one, then the other, and you try to describe what you see without letting your hopes write the answer. You start with simple observations. Color in bulk. One looks darker, more organic. The other looks lighter, more mineral. Grain sizes. One has more sand, the other more silt. Mineral sparkle. One has more mica plates, flashing as you tilt the light. Dark, heavy grains. One has more opaque particles peppered through the field. Then you look for the unusual bits. Does either have pollen that stands out? Does either have perlite granules? Does either have spherical fly ash particles? Does either have a distinctive fiber color? You write it down. You do not trust memory, because memory is a liar that thinks it is helping. And as you do this, you realize why forensic reports often sound boring. They are boring on purpose. Boring is what careful sounds like. Now you let the microscope linger on a single grain, a quartz chip with tiny fractures. Those fractures could have formed during weathering, or during crushing, or during transport. They look like a network of fine lines, like a cracked windshield in miniature. And you remember that every grain has been through something. Geology is trauma, stretched over time. Now you shift the stage and catch a mica plate. It is thin, reflective, and it changes brightness as you move the light. Mica cleaves into sheets because its crystal structure is layered. Those layers split easily, producing the thin plates you see. Mica in soil can hint at source rocks, like metamorphic rocks or certain igneous rocks. It can also show up in many sediments because it survives transport reasonably well, though it can break down. If you see abundant mica, you might suspect a source region with mica-rich rocks or a sediment derived from them. You might also think about how mica affects soil behavior. Mica plates can influence texture and water holding, and in large amounts, they can contribute to a shiny look in soil. Then you drift to a feldspar grain, slightly cloudy. Feldspars are common, but they weather into clay more readily than quartz. So, a soil rich in feldspar might suggest relatively young sediment, or less intense weathering, or continuous replenishment from nearby rock. In some environments, like arid regions, feldspar can persist longer because chemical weathering is slower. In humid regions, feldspar tends to alter more quickly. So even the presence or absence of certain minerals can hint at climate indirectly, not because the grain knows climate, because chemistry does. Now, if your microscope setup allows, you might do a simple hardness test on larger grains outside the slide, or a basic acid test on bulk soil. A drop of dilute acid can fizz on carbonates, like calcite from limestone. That fizz is CO2 being released as the carbonate reacts. If a soil fizzes, it suggests carbonate content. That can be natural, from limestone bedrock, or shell fragments, or carbonate-rich dust. It can also come from concrete dust in urban areas. So again, you interpret with context. Everything in soil can be natural, human, or both. Now you begin to feel the rhythm of comparison and the calm reality that it is rarely about perfection. If two soils are identical in every way you can measure, that is unusual. More often, you find they are similar in many ways and different in a few. So you ask, are the differences explainable? Could one sample have more plant fragments because it was collected closer to surface? Could one be lighter because it dried more? Could one be missing fine particles because rain washed them off? Time matters. Weather matters. This is why sample collection is so important. A good forensic comparison begins long before the microscope, with someone at a scene collecting soil properly. A careful collector will document location, depth, and conditions. They may collect multiple control samples from different points around a scene because soil can vary over short distances. They will use clean tools, avoid cross-contamination, and store samples in clean containers, often paper packets for soil, so moisture can escape and mold does not grow. If you seal wet soil in plastic and forget it, biology will throw a party. Mold can grow, chemical conditions can change, and the sample can alter. Paper breathes. It lets the soil dry slowly, preserving it more faithfully. So even the container matters. In the lab, you can often tell if a sample was handled poorly. Mixed debris, obvious contamination, inconsistent labeling. That is when skepticism becomes your best tool. Now, speaking of skepticism, you start thinking about what soil cannot promise. Soil can suggest association. It can suggest consistency. It can sometimes exclude. If samples are clearly different, but it rarely provides individualization in the way fingerprints or DNA can. Many locations share similar geology. Many urban soils contain similar mixtures of construction debris. Many regions have widespread wind-blown dust that blankets everything with the same fine particles. So when someone asks, "Can you say this soil came from this exact spot?" the honest answer is usually no. You can say it is consistent with that spot, and you can evaluate how common that consistency is. That evaluation requires background knowledge, reference samples, databases sometimes, experience with regional soils. It is not glamorous. It is methodical. And because it is methodical, it has limits that must be respected. One major limit is sample size. Sometimes you have almost nothing. A few grains in a seam, a smear on fabric. In those cases, you must decide what tests are worth using, because every test consumes material. Another limit is heterogeneity. Soil varies. Even within a small area, you can have patches of different soil types, or different degrees of organic matter, or different human debris. If you collect one control sample and assume it represents the whole scene, you can mislead yourself. Another limit is time. Soil changes, it can dry, crack, oxidize, grow mold, lose volatile components, be washed by rain, be mixed by footsteps. A sample collected weeks later may not match a sample collected at the time of an event, even if they came from the same place. Then there is contamination, the eternal nuisance. Soil transfers easily. If evidence is stored poorly, if tools are reused without cleaning, if samples are open near each other, particles can move. And because soil is made of tiny particles, you might not notice contamination until you compare and wonder why everything looks vaguely similar. This is why lab practices matter. Separate work areas, clean tools, careful labeling, working one sample at a time, boring procedure, saving you from exciting mistakes. Now, because this is a bedtime story for the existentially curious, you allow yourself a philosophical pivot, gentle and quiet. Soil is time, made granular. It is rock that lost its certainty. It is leaf that lost its shape. It is the residue of things that were once alive and now feed what is alive. It is a constant recycling system that does not care about your plans. In forensics, you ask soil to help you reconstruct human moments, short moments measured in minutes or hours. But soil itself is indifferent to those moments. It measures in seasons and centuries. It sits through your urgency like a patient older than your whole species. And that is oddly calming. Because if the ground can hold endless detail and still remain quiet, maybe you can too. Now, you return to the microscope and let it offer you something that is not just useful, but strangely beautiful. You switch back to polarized light again, not for identification this time, but for the slow color shift. You rotate the stage, and grains flicker through interference colors, as if they are responding to a secret signal. The colors come from how polarized light splits and recombines as it passes through birefringent crystals. The amount of color depends on thickness, orientation, and the mineral's birefringence. In a thin section of rock, geologists use these colors for identification, along with extinction angles and twinning. In loose soil mounts, it is less controlled, but still informative. You watch one grain go dark, then bright, then dark again. As you rotate, that is extinction when a crystal orientation blocks the polarized light. It is a simple rhythm, bright, dark, like blinking, but slower. Then you see a grain with faint stripes. That could be feldspar twinning, a pattern in the crystal structure. It looks like tiny parallel lines, delicate and internal. You might not name it confidently tonight, but you notice it, you remember it, and that is how your mental library grows. Now you move the slide again and catch a tiny carbonate fragment. Under polarized light, calcite can show strong birefringence and vivid colors. It can also show cleavage, little straight planes. If you had a drop of acid, it would fizz. Here, it just glows and sits there. Carbonate fragments can come from limestone bedrock, shell debris, or urban concrete dust. Context decides which interpretation is sensible, and context is the quiet king of forensics. Now your attention drifts to a different kind of microscope view, one that some labs use for soil comparisons because it preserves surface detail better. Stereomicroscopy. A stereomicroscope gives you a three-dimensional view at lower magnification. It is the microscope you use when you want to see the overall texture of grains, their shape, their luster, their color, without flattening them under a cover slip. Under a stereo microscope, you can pick out individual grains with tweezers. You can separate a suspicious fiber. You can isolate a paint fleck. You can sort by color or size. So the process often moves between instruments: stereo microscope for sorting and selecting, compound microscope for detail and optical properties. If you had both in front of you, your work would become a gentle routine. Sort, mount, view, note, compare. The calmest kind of loop. Now, you let your mind consider how this information is presented in real cases. A forensic soil examiner may write a report describing the questioned sample and the known sample, listing characteristics and indicating whether they are consistent. They may use terms like cannot be excluded as originating from, or exhibit similarities consistent with. Those phrases sound cautious because they are. They are built to avoid overstating. In court, that caution can sound less satisfying than certainty. People want yes or no, but soil does not give you yes or no. Soil gives you a stack of probabilities, and your job is to stack them honestly. Now you shift again, because there is something you have been circling but not fully entering yet. The tiny hitchhikers. You have seen hints of pollen and fibers, but you have not lingered in their strange shapes the way they deserve. So you prepare another slide, deliberately choosing a bit of the sample that looks darker and more organic. That is often where biological particles hide. Mineral-heavy sand can dominate a view and make it harder to spot pollen. Organic rich fractions can concentrate it. You mount it gently, lowering the cover slip slowly to avoid crushing delicate structures. Then you focus. At first, you see the familiar chaos of fragments. Plant tissue bits, dark clumps, fine particles. Then you see it. A pollen grain caught in the field like a tiny object from a different world. It has a textured outer wall with a pattern of ridges. It is not perfectly smooth. It looks engineered, which it is. Plants evolved pollen walls to survive ultraviolet radiation, dryness, mechanical stress, and the general cruelty of transport. Wind can carry pollen for kilometers. Insects can carry it on hairs and legs. Water can move it too. So pollen has to be tough. Under higher magnification, you might see apertures, little pores or furrows where the pollen tube can emerge during fertilization. The number and shape of apertures can help classify pollen. Some have one furrow, some have three, some have multiple pores. You do not need to memorize this tonight. You just need to appreciate that a plant's reproductive cell comes with its own architecture. And in soil, that architecture becomes evidence. If you find a pollen type associated with a specific plant community, it can suggest habitat. If you find pollen from a plant that is seasonal, it can suggest timing. If you find a mix of pollen types, it can suggest the surrounding vegetation, like a blurry botanical snapshot. But you also remember the limitation. Pollen can travel far. Wind-pollinated trees release huge amounts. Their pollen can dominate, even if the tree is not right next to the soil source. So you interpret carefully. Then you see another pollen grain. spiky. It looks like a tiny medieval weapon. Some pollen types have spines, often insect-pollinated plants. The spines can help pollen stick to pollinators. So, spiky pollen may suggest different plant groups than smooth, wind-carried pollen. Again, suggest, not guarantee. Now you let the microscope drift to a fiber, pale blue, smooth, and uniform. It lies across the field like a rope across boulders. It is clearly not mineral. It is too consistent. You adjust focus up and down, and the fiber's edges sharpen. You look for surface features. Does it show twists like cotton? Does it show scales like wool? Or is it smooth like many synthetics? It appears smooth. You note it as a synthetic fiber, blue, consistent diameter. Then you look for dye granules or internal structure, but you do not push too hard. This is a sleep-friendly journey, not a full lab training. You move on. Now you spot a tiny flake, reflective with a colored surface. It could be mica. It could be paint. Under bright field, it is hard to tell. Under polarized light, mica would show birefringence and a certain behavior. Paint may not. You switch briefly, observe, and it stays mostly unchanged. That suggests it might not be a crystalline mineral. You note it as a possible paint flake or synthetic fragment. You do not declare victory. You do not write a story. You write a note, and this is how you build comparisons. When you have a known soil from a location, you look for the same kind of flake, same color, similar thickness, similar abundance. If both samples share it, that is another small agreement. Now, you let your attention broaden again, because the next step in the outline of your night is the gentle art of comparing samples more deliberately, more methodically. So you imagine two small piles of soil like two bowls of spices on a kitchen counter. They may both be brown. They may both smell faintly of earth, but one has more sand crystals that catch light, and the other has more fine powder that clumps. This is where a lot of examiners begin. Before microscopy, simple physical observations. Color can be described with standardized charts, like the Munsell soil color chart, which gives a structured way to describe hue, value, and chroma. That helps reduce subjective language. Instead of saying reddish-brown, you assign a code. Texture can be estimated by feel, though this is more common in soil science than strict forensic work. Still, the basic impressions matter. Gritty suggests sand, smooth suggests silt, sticky suggests clay. Smell can matter too, though it is subjective. A soil rich in organic matter can smell more earthy. A soil with petroleum contamination can smell like fuel. A soil with certain industrial contaminants can have distinctive odors. You do not rely on smell alone, obviously, but you note it if it is strong. Clumping behavior can be informative. Does the soil form hard clods when dry? Does it smear when wet? Does it crumble easily? Then you move to microscopy for the details. You compare slides from sample 1 and sample 2. You count, roughly, how many grains in a field are clear quartz, how many are dark opaque, how many are shiny mica plates. You do not need exact counts for every case, but systematic observation helps. Some labs do semi-quantitative counting, like point counting. You view a field and tally grain types at regular points. This can give proportions. Proportions are useful because they are harder to argue with than vague impressions. You might also separate samples into size fractions with sieves and compare each fraction. Sometimes differences hide in a specific size range. For example, the fine fraction might contain most of the clay and organic matter, and therefore most of the coatings and biological particles. The coarse fraction might show distinctive mineral grains or industrial fragments. So you compare fraction by fraction. Again, not glamorous. Effective. Now, a quiet but important point. When you compare, you are not just looking for similarities, you are looking for differences too. It is tempting to focus on what matches, but differences can exclude. If one sample contains abundant carbonate shell fragments and the other contains none, that is a meaningful difference, depending on context and sampling. If one sample contains a distinctive industrial particle, like spherical fly ash, and the other has none, that might matter. But you also ask, could the difference be due to sampling? Did one sample come from a wetter area? Did one include more surface litter? Did one get washed by rain? This is why multiple control samples are valuable. If controls vary widely, then a difference may not be meaningful. If controls are consistent and questioned sample differs, then the difference becomes more significant. The strength of soil evidence often comes from good sampling strategy. Now, you let yourself consider a case-style scenario, gentle and non-graphic, because the details do not need to be grim to be real. A vehicle is suspected of having driven into a rural area. Soil is found in the wheel wells. Investigators collect soil from those wheel wells, and they collect soil from the suspected rural track. Under the microscope, both show a similar mix of quartz and feldspar. That is common. But both also show abundant mica plates, and both contain small fragments of a specific red brick-like material, likely from a broken culvert or nearby structure. Both contain spherical fly ash particles, suggesting proximity to a road or industrial source. The combination of features becomes more informative than any one feature. So the conclusion becomes, the samples are consistent, and the combination of observed features is not extremely common in the wider region, based on available knowledge. That is how soil evidence supports an investigation. Not by being dramatic, but by being additive. Now you return to your bench, and you feel the quiet satisfaction of process. You have a stack of notes, plain language, no poetry, more shiny grains, more dark fragments, presence of blue synthetic fiber, occasional spiky pollen. Fine coating suggests clay. This is the part of forensic science that feels almost like accounting. And that is a compliment, because in a world that loves spectacle, the truth is often assembled by people who are willing to be bored on purpose. Now, since the world is messy, you also learn the gentle limits again, in a slightly different form. Even with careful comparison, soil can rarely be said to originate from a single point. It can support a claim of association between an object and a location. It can help reconstruct movement. It can sometimes contradict a story. But it should not be oversold. So you imagine the courtroom version of this, the one where someone demands certainty. The examiner calmly explains that soil evidence is class evidence, not individual evidence. It can classify and associate, but not individualize. That honesty is the difference between science and theater. Now, to keep your mind from getting too tense, you let it drift into the strangely beautiful side of microscopy, the part that is almost meditative. You pick a field with a mix of grains, and you simply watch how light interacts with them. Under bright field, some grains sparkle. Under polarized light, some glow. Under oblique light, surfaces show relief. You focus up and down slightly, and the scene changes like fog lifting and settling. A grain comes into crisp view, then fades as you move to a different depth. The mounting medium has thickness. The grains sit at different heights, so focusing becomes a slow exploration. Blur, sharp, blur. And as you do this, you begin to realize that microscopic beauty is not an exception in nature. It is the default. We just do not have the optics to see it most of the time. The universe hides detail behind scale, like a miser hiding coins. Soil is especially good at hiding detail because it is literally the leftovers of everything. It is geology and biology and human debris blended together. And under a microscope, it becomes a collage. You linger on a crack in a grain that looks like a dried riverbed. You think, briefly, about how rivers carve landscapes at large scale the same way fractures carve stress patterns at small scale. Physics repeats itself across sizes because it is efficient and uncaring. Then you notice a smooth grain surface that looks like a frozen lake. You think about water again, shaping rock slowly, then leaving behind its traces. Time is a sculptor that never takes a day off. Now you let your mind drift toward the odd comfort in that your worries feel urgent because your brain is built for urgency. But the ground beneath you has been changing for longer than your species has existed. It will keep changing long after your to-do list stops mattering. That is not dismissive. It is just scale. So, back at the bench, you continue comparing, but you do it with the patience of geology. You pick a new area on the slide, and you scan for rare inclusions. You spot a small piece of charcoal, black and porous. Charcoal can persist in soil for a long time. It can come from wildfires, controlled burns, fireplaces, industrial emissions. Under magnification, charcoal often shows a woody structure, little pores, and channels. If charcoal is abundant, it can suggest a history of burning in the area, though again, context matters. You spot a tiny shell fragment, pale and curved, with a layered look. That suggests carbonate material, possibly from a marine environment or from crushed limestone used in construction. Or road base, you spot a particle that looks like a tiny bead, perfectly spherical. You note it again as possible fly ash. Then you spot a translucent particle with bubbles inside, like tiny voids. That could be a volcanic glass fragment, or a lightweight aggregate, or something else. If your region has volcanic activity, volcanic ash can contribute glass shards. In urban settings, lightweight aggregates used in construction can mimic that look. So, you do not jump. You simply note it as a vesicular, bubble-rich particle. And you see how forensic soil work is often about resisting the urge to be certain too quickly. Now, the quiet truth is that many labs use additional analytical methods beyond optical microscopy when needed. They might use X-ray diffraction to identify clay mineral types, because clay minerals are too small to identify easily under a light microscope. X-ray diffraction looks at how crystals diffract X-rays, producing patterns that correspond to specific mineral structures. They might use infrared spectroscopy to identify organic materials, or certain synthetic fragments. They might use scanning electron microscopy with energy dispersive spectroscopy to look at particle composition, telling you what elements are present. Those tools can provide more objective identification. But even then, the story stays the same. You compare, you evaluate consistency, you consider how common the observed combination is. Optical microscopy is often the first step because it is fast, relatively simple, and very informative. It is also conveniently quiet. Now you feel the natural bridge approaching because you have been circling around the same idea. Ordinary details become meaningful when you compare them carefully. So you keep leaning into that sleepy art of comparison, letting it become the center of your attention. You imagine a reference collection, shelves of little labeled containers from different locations, soil from parks, roadside verges, riverbanks, forests, construction sites. Each one looks boring until you look closely. In some cases, investigators build local soil databases, mapping soil characteristics across regions. Soil surveys exist too, created for agriculture and land management, describing soil types, textures, and distributions. Those surveys can provide context, though forensic questions often require more fine-grained comparisons. Because a soil map might tell you a general soil series in an area, but not the human debris, the pollen mix, the tiny industrial signatures that make a sample distinctive. So you still end up at the microscope, watching grains like tiny planets. You compare one more time, sample A and sample B. You notice sample A has more clay coating, giving grains a slightly stained halo. Sample B has cleaner grains, suggesting more washing or less developed soil structure. Sample A has abundant organic fragments. Sample B has fewer. Then you find something that makes you pause, not with excitement, but with that quiet feeling of pattern recognition. Both samples have the same unusual green fiber, thin, smooth, and slightly reflective. Not common, not rare, but distinctive enough to note. Both samples have small brick-like red fragments. Both show the same general mineral mix. This is how soil evidence becomes persuasive, not by one miraculous particle, but by several small agreements that are harder to dismiss together. But you also remember to ask, could those agreements be common in the environment? Green fibers might be common if the area has synthetic turf or certain textiles. Brick fragments might be common in older neighborhoods. So you do not overstate. You simply stack what you know and leave space for what you do not. Now you let the microscope drift again, because you have been doing serious work and the night likes softness. You watch the grains shimmer under polarized light. You rotate the stage slowly, and it looks like the field is breathing. Colors brighten and fade, crystals go dark and light again. It is almost unfair that something as grimly practical as forensic evidence can also be this pretty. But nature does not care about your categories. Useful and beautiful can be the same thing by accident. Now, without rushing, you find yourself returning to a practical question, one that examiners quietly answer all the time. How much weight should soil evidence carry? The answer depends on the case context and the quality of sampling and analysis. Soil evidence is strongest when it includes a combination of features that are not extremely common, and when sampling is good, and when alternative explanations are considered. It is weaker when soils are very common, when samples are tiny, when contamination is possible, when controls are limited. So you keep the mindset of patient skepticism. You treat soil as a supportive witness, not the hero of the story. That is how you avoid becoming a storyteller instead of a scientist. And speaking of what soil cannot promise, there is a gentle relief in admitting limits. Limits mean you stop arguing with reality. Limits mean you work inside what is true. So you let that thought sit on the bench beside the microscope, quiet and solid. Then you turn back to the slide, because there is still more to notice, and the night is still young enough to keep looking. You let the stage drift just a few millimeters, and the field changes completely. A whole new neighborhood of grains slides into view as if the previous scene never existed. That is one of the quiet lessons soil teaches you. Your certainty is always one gentle nudge away from being wrong. And that matters, because soil is not cooperative. It is not a clean substance with a clean answer. It is a crowd, and crowds are statistical. So when you compare samples, you start treating your microscope like a counting machine, not a theater. You pick a simple rule, something boring and repeatable. You scan left to right. You stop at regular intervals. You note what is under the crosshair or at the center of the field without favoritism. Quartz, clear and stubborn. Dark opaque, unhelpfully anonymous. Mica plate, flashing like it wants attention. Organic fragment, dark and soft-edged. Possible pollen, patterned and too intentional to be rock. You do this because your brain loves to cheat. Your brain will remember the rare, glittery thing and forget the 80 plain grains that actually define the sample. You are trying to keep the romance out of it. Congratulations, you are babysitting your own perception. Now you glance at your notes and you notice how plain they look. More clear grains, fewer dark fragments, some coatings, occasional fibers. It reads like someone describing the contents of a vacuum cleaner, which is honestly what forensic soil is: vacuum cleaner archaeology. And this is where the limits become practical, not philosophical. Because when someone asks for a match, what they often mean is a promise. They want you to say the soil came from one exact patch of ground, like the universe bothered to label it. But soil distributions do not behave like that. They overlap, they repeat, they smear into each other. So you begin thinking in questions that soil can actually answer. Are these samples consistent in mineral mix? Are they consistent in grain size distribution? Do they share uncommon inclusions, the tiny extras that narrow the world? And maybe most importantly, are there differences that cannot be ignored? You let your eye settle on a single bright grain, and you feel how easy it would be to overinterpret it. Maybe it is glass. Maybe it is quartz. Maybe it is a synthetic fragment. Under bright field, it is tempting to just decide. So you switch the lighting again, briefly, and watch how it behaves. Under crossed polarizers, quartz will usually show that low gray to white interference look. Glass, being amorphous, often stays dark or behaves differently because it does not have the crystal structure to split light in the same way. You do not need to be dramatic about it. You just watch, rotate, and note. If it stays dark through rotation, you consider glass, or a non-crystalline fragment, or an opaque particle. If it brightens and dims in a predictable way, you lean back toward crystalline minerals, not certainty, just direction. Now, while you are still in this careful mood, you let yourself think about what happens before the soil ever reaches you. Because the microscope is only as honest as the collection. At the scene, someone has to decide where to take controls, not just one scoop, not just the most obvious spot. They collect multiple because soil changes over small distances. A few steps toward the shade, more organic material. A few steps toward the gravel path, more crushed stone. A few steps near a building, more construction dust. So good sampling looks boring and redundant. Many small envelopes, many labels, many photos. And every envelope is a tiny decision about what story you will be able to tell later. In the lab, you can sometimes feel the difference immediately. A well-collected set of samples comes with context, depth, location, separation. A sloppy set comes like a riddle with half the words erased. And when you get half a riddle, you are not allowed to invent the missing parts. Now you find yourself thinking about contamination again, but in a more physical way. Soil particles are small, and they behave like dust. Because they are dust, they cling to gloves. They float when poured. They ride on static charge. So in a real lab, the quiet rituals are not just tradition, they are defense. You open one packet at a time. You keep lids and flaps closed. You clean the bench between samples. You use fresh paper. You change gloves when you need to. You do it because you have seen what happens when you do not. Everything starts to look consistent because you accidentally made it consistent. Congratulations! You just forged evidence with your own hands, and you did not even mean to. So you keep your movements slow, and you keep your workspace small. One slide, one sample, one set of notes. And when you compare two samples, you compare them like a person who expects to be wrong the first time. Now, somewhere in the middle of all this restraint, you notice something quietly satisfying. Soil evidence is not about a single perfect identifier. It is about combinations. A quartz-rich sand alone is common. A quartz-rich sand with abundant mica plates, a few spherical fly ash particles, and a consistent set of brick-red fragments, that combination starts to feel more local. Still not unique, but more informative. So you let your mind build a picture, the way it always tries to. But you build it with handrails. A place with quartz and mica suggests certain source rocks or sediments derived from them. The brick-red fragments suggest human construction debris. Nearby, maybe older masonry. The fly ash suggests combustion sources, traffic corridors, industrial emissions, or even distant transport depending on region. That is already a more specific environment than somewhere on Earth, which is the most soil will ever promise you. Now your eyes drift down to something you have been ignoring because it seems too simple. The soil on the slide is not evenly spread. There are small clumps and thin areas and little islands of fine material. That distribution itself is information. Fine particles like clay and organic fragments tend to clump because electrostatic forces and moisture make them cling. Sand grains are heavier and tend to sit where they are dropped. If you see fine material coating sand grains, you suspect clay content, or organic staining, or both. And when you see a clump, you remember that soil often travels as aggregates, little packets of mixed material held together by chemistry and biology. In a way, aggregates are the soil's preferred form. They are more stable, they resist erosion a bit better. They create pore spaces for air and water. So the presence of stable aggregates can hint at a biologically active soil, with microbial glues and root exudates helping hold it together. A highly disturbed soil, constantly tilled or trampled, might show more broken aggregates and more loose particles. That is not a moral judgment. It is just what physics does when you stomp on things. Now your mind pivots gently because you realize you have been living in mineral land for a long time. And mineral land is patient. Biology is not. Biology is more seasonal, more local, more specific. And that is why those tiny hitchhikers matter so much. So you return to pollen, but you approach it like a careful listener, not a collector of trivia. Pollen is everywhere, but not evenly. Wind-pollinated plants, like many trees and grasses, dump massive amounts into the air. Their pollen becomes background noise, the static of the landscape. Insect-pollinated plants often produce less pollen, but it may have more elaborate surfaces because it is designed to stick to bodies. So when you find pollen in soil, you ask what kind of pollen dominates. Is it mostly the common wind stuff, or do you see distinctive grains that suggest local flowering plants? You slide to a new area and find a grain with two air sacs like tiny ears attached to an oval body. That kind of shape is common in certain conifers. The air sacs help the pollen float and drift and invade your sinuses like it owns the place. You note it, possible conifer pollen. Then you find a smaller grain with multiple pores, a kind of dotted pattern. It could be from grasses, which can be difficult to identify beyond family with basic microscopy. Still, grasses tell you something. They suggest open areas, lawns, fields, roadside vegetation. You do not need the plant's full name to let it contribute to the picture. Now you drift to spores, because soil is basically a spore storage facility. Fungal spores can be round, oval, sometimes with distinctive ornamentation. They can be dark, especially if pigmented to resist sunlight. They can be abundant in soils rich in decomposing organic matter. If a soil sample has many fungal spores and lots of plant fragments, you suspect a more organic environment. Leaf litter, garden beds, forest floor, compost-rich soil. If it has fewer, you might suspect more mineral-dominated environments, or simply a different season, or a different microhabitat. Again, suggest, not promise. The constant theme, like a lullaby you did not ask for, is that the world is messy and your job is to be honest about it. Now, as you stare at these biological particles, you remember another quiet limit. Biology changes faster than minerals. A soil collected in summer may have different pollen and spore content than the same soil collected in winter. A rainstorm can wash away some surface pollen. A windy week can deposit more. So when pollen helps, it helps best when the timeline is tight and the sampling is good. Otherwise, it becomes a beautiful hint that refuses to become a verdict. Now you pivot again to something that feels almost too modern to be called soil evidence. Microplastics. You might see them as colored fragments or thin films or fibers that are too smooth, too uniform, too perfect. Sometimes they look like tiny, translucent flakes. Sometimes they look like bright shards that do not behave like minerals under polarized light. Microplastics are everywhere now, including places that should not have them. They travel by air and water. They settle into soils and sediments. They become part of the background. So in a forensic context, microplastics can be a clue, but often they are just there, like an unwanted watermark on the entire planet. If you find a very distinctive plastic fragment, a specific color and shape, maybe with embedded glitter, maybe with a layered paint-like structure, it can matter. If you find generic microplastic fibers, it may not, and you learn quietly to tolerate that ambiguity. Now your mind drifts back to the act of comparison, because that is where all of this becomes practical. You imagine a question soil sample from a shoe and a control sample from a location. You compare them under the microscope, but you also compare them as bulk samples, the way soil scientists do. Color is one, and color can be surprisingly complex. A reddish soil often suggests iron oxides, like hematite, coating grains. A yellowish or brownish soil may involve different iron oxide forms, like goethite. Gray soil can suggest reduced iron conditions, often in waterlogged environments, where oxygen is limited and iron chemistry changes. Black soils can be rich in organic matter or influenced by charcoal, soot, or industrial carbon. White or pale soils can be high in carbonates, gypsum, or simply sand with little coating. So color is chemistry, but it is also lighting, moisture, and how the sample was stored. Dry soil looks lighter, wet soil looks darker. Fine particles can make soil look darker because they coat surfaces. Organic content darkens everything like a stain. So, if you compare color, you compare it under consistent conditions, similar drying, similar lighting, preferably with a standard reference like a color chart. You do not trust your late-night eyes alone. Late-night eyes will call everything brown enough. Now, beyond color, there is a simple test that soil scientists use that has a quiet forensic relevance too. The sedimentation idea, how particles settle in water. If you put soil in a clear container with water and a dispersant, then shake it and let it settle. The largest particles settle first. Sand drops quickly. Silt settles slower. Clay can stay suspended for a long time, making the water cloudy. This is basically Stokes's law in a jar, but you do not need the math tonight. You only need the image. Bigger particles fall like stones. Tiny particles hang like a fog. So a quick settling test can give you a rough sense of texture differences between samples. One jar clears quickly, sandy. Another stays cloudy longer, clay-rich. In strict forensic contexts, you would document this carefully, and you would use standardized methods, but the principle remains soothingly simple. Gravity sorts, and time reveals the sort. Now you glance back into the microscope and see a grain that looks like it has tiny grooves, almost like teeth marks. You wonder, briefly, if it is a mineral with cleavage or a fragment of something manufactured. You could chase it, but you decide to do what good examiners do. You categorize it and move on, unless it repeats. Because a single weird particle can be noise. A repeating feature becomes signal. So, you keep scanning until you see another similar fragment. Then another. Now you pay attention. If you see repeated fragments of a specific type, like crushed glass, or brick, or shell, or asphalt, it begins to define the sample. Repetition is how soil speaks louder. Now, because your mind likes stories, you let it build a gentle case study that stays non-graphic. You are asked to compare soil from a bicycle tire to soil from a path near a canal. The tire soil has fine silt, some clay coating, and scattered diatoms. The control soil from the canal path shows the same fine texture, the same kind of clay film, and also diatoms, including a distinctive long needle-like form. A dry upland path might have fewer diatoms. A sandy beach might show different mineral sorting. A garden bed might have perlite and fertilizer granules. So, the presence of diatoms plus the texture supports contact with a wet environment consistent with the canal path. It does not prove the bicycle was at that exact meter of ground, but it supports a story of movement that includes that kind of place. Soil evidence does not shout, it leans. Now, with that thought in your pocket, you return to the microscope's quiet beauty again, because your brain can only do careful skepticism for so long before it wants something softer. You switch to polarized light again and let the grains shimmer. Mica plates flare bright, then disappear as they rotate into extinction. Quartz sits there with its modest gray. Feldspar may show faint internal patterns. Carbonates can blaze with color. It is like watching a slow, silent dance where every partner follows rules older than language. And in the middle of that dance, you notice something that feels almost like a secret. Under crossed polarizers, many man-made materials behave differently than crystalline minerals. Glass tends to stay dark because it is amorphous. Many plastics also behave differently, sometimes showing stress patterns if they are under tension, sometimes showing strange internal birefringence if molded or stretched. So polarized light can sometimes help you separate the natural from the manufactured, gently, without needing to destroy the particle. That can matter in urban soil, where the mineral mix includes crushed construction materials that are not minerals at all, but still behave like hard grains. Now your mind returns to the limits again, but this time with a practical softness. If you have two samples that are broadly similar, that might simply reflect regional geology. Many places share quartz, feldspar, and mica. Many places share similar road dust. So what makes a soil comparison stronger is not the common backbone, it is the uncommon garnish. A specific pollen assemblage, if stable, a distinctive industrial particle mix, a unique combination of brick, paint, and fibers. A particular heavy mineral suite, if you can characterize it, like an abundance of magnetite, garnet, or zircon, depending on region. Heavy mineral analysis is a whole quiet world of its own. Heavy minerals are the denser grains that can be separated from soil using heavy liquids in specialized labs. They can provide a more distinctive fingerprint because certain source rocks produce certain heavy mineral mixes. But heavy mineral work is more involved and not always necessary. Often, the simpler optical comparisons and inclusion observations are enough to support or exclude. And for sleep-friendly purposes, it is enough to know that soil has layers of analysis, and each layer costs time, effort, and sample material. So, you choose your layer based on the question. Now, since your question tonight is basically, how does forensic soil microscopy work without becoming nonsense, you keep your focus on the calm heart of it. Comparison is a craft. It relies on consistent viewing conditions, careful notes, repeated observations, and humility about uncertainty. It also relies on something that sounds unscientific until you do it enough. Pattern recognition built from experience. An examiner who has looked at hundreds of local soils develops a sense of what is common in that region and what is unusual. That does not replace measurement, but it guides attention. You learn where to look. You learn what is likely to matter. And you also learn what not to chase, like a single glitter fleck in a world that has been covered in glitter since the late 20th century. Now you feel the night deepen outside the lab windows, and the building remains indifferent. Ventilation hums. A distant pipe ticks as it cools. You decide to do one more kind of check that is quietly important, the idea of exclusion. Because sometimes the most useful thing soil does is tell you no. If a questioned soil is coarse beach sand, full of rounded quartz shell fragments and salt residues, and the control soil is clay-rich forest soil with leaf litter and fungal spores, you do not need sophisticated analysis to say they are inconsistent. If the questioned soil contains abundant gypsum and drywall dust, and the control is natural river silt without those materials, you note the mismatch. Exclusion can be cleaner than inclusion because differences can be obvious, but you still stay careful because mixtures exist. A shoe can carry both beach sand and garden soil layered in the tread. So even exclusion requires you to consider whether you are comparing the same fraction, the same layer, the same part of the tread. This is why some soil work involves taking multiple subsamples from the same item. Heel versus toe, outer tread versus inner tread, deep groove scrapings versus surface dust. You are not just comparing soils, you are comparing time layers. And time layers are exactly what soil is made of. Now your gaze settles on the edge of the cover slip, where particles sometimes accumulate. Surface tension during mounting can pull fine material to edges, like a tide line. If you forget that, you might think the sample naturally has a band of fine particles. So you note it, you interpret cautiously. Even slide preparation has artifacts. Bubbles can mimic round particles. Mounting medium can refract light strangely. Crushing can break delicate structures. So you develop the quiet habit of asking, could I have caused this? That habit is the difference between observation and self-deception. Now you let yourself wander gently into the strange beauty chapter without calling it that, just letting it happen. Because there is a moment when you stop trying to extract meaning and you simply watch patterns exist. A quartz grain shows internal fractures like a spider web. Frozen stress. A feldspar fragment shows faint cloudy alteration, like bruising from chemistry. A mica plate reflects a clean flash, then disappears. A dark organic flake sits like a charred leaf in miniature. A pollen grain rests among them like a tiny alien egg, all geometry and purpose. And the absurd part is that this is just the ground. This is what you walk on. This is what collects in the corner of your hallway. This is what ends up in your bed sheets if you do not take your shoes off. The world is not hiding wonders. You are just too large and too busy. Now, speaking of busy, you remember how long it takes nature to make these grains. Quartz crystals form in igneous rocks, cooling from melt or precipitating from fluids, and then they sit there for millions of years until weathering breaks them free. Feldspar alters into clay slowly through water and carbonic acid. Mica splits into plates along crystal layers. Organic matter enters as leaves and roots, then gets shredded by insects, bacteria, fungi. Humus forms, complex and dark, storing carbon and nutrients. So every soil grain is a survivor of multiple processes: heat, pressure, water, biology, transport. And then a human steps on it and calls it dirt and wipes it off on a doormat. Congratulations! You are the briefest chapter in an extremely long book. Now, you tilt the stage slightly and watch how the light changes. Under oblique lighting, shadows stretch. Tiny ridges stand out. A grained surface looks like it has been sandblasted. You think of wind again, patient and picky, sorting sand in dunes, frosting surfaces with tiny collisions. In some deserts, sand grains collide so often that they become rounded and matte, and sometimes even develop tiny crescent-shaped pits. Under a light microscope, you may not see every microtexture, but you can sometimes sense the difference between a grain that has been rolled in water and one that has been battered by air. Water polished can look smoother, more glossy. Wind frosted can look more matte, more textured, and that sense can help you imagine the environment that produced the soil without claiming to know it with certainty. Now, you let your mind slow into that geologic patience again. In the macro world, landscapes change slowly. In the micro world, your focus knob changes landscapes instantly. That contrast is part of what makes microscopy feel like a small power. You get to command scale, but the microscope is also a humbling device because it shows you how much is always there, unnoticed. Now you find a small cluster of particles that look oddly uniform, almost like crushed white foam. You adjust focus, and the surfaces look porous, full of holes. Perlite, you think? That common potting soil additive? Expanded volcanic glass, heated until it pops like popcorn, leaving a lightweight porous structure. Perlite in soil can suggest gardening, landscaping, potted plants, or soil amendments. It is not natural in many soils at that concentration. So if you find it in a question sample, and also in a control sample from a garden bed, that can be a useful point of consistency. And it is such a small domestic detail, the kind of clue that comes from someone trying to keep a houseplant alive. Forensics often lives in the overlap between the cosmic and the mundane. Now you drift to another domestic intruder, fertilizer granules. They can appear as small, rounded pellets, sometimes with a crystalline look, sometimes with a coating. If they are fresh, they may have sharp edges. If weathered, they may be partially dissolved. Fertilizer presence can suggest cultivated soil, lawns, gardens, agricultural areas. Again, not proof, just context. And the context matters because it helps you avoid making the soil sound more mysterious than it is. Most soil clues are ordinary. They become meaningful because you notice them carefully. Now your notes are getting fuller. You have the backbone mineral mix. You have biological hitchhikers. You have human debris hints. You begin to see how a report could be written from this, quietly and honestly, not as a verdict, but as an assessment. The question soil exhibits a mixture of quartz, feldspar, and mica. It contains moderate organic material. It includes occasional pollen grains consistent with local vegetation and rare spherical particles consistent with fly ash. It contains white porous fragments consistent with perlite. The known soil from the location exhibits a similar mixture and similar inclusions. Therefore, the questioned soil cannot be excluded as having originated from that location and is consistent with contact. That kind of language is careful. It gives the court or investigators something real without pretending soil is a fingerprint. Now you let your eyes rest again on the shimmering field and you realize the weird truth. This work is soothing because it is slow. Everything else in life wants quick answers. Soil refuses. Soil demands patience. And in exchange, it gives you small truths. Now, as the night moves forward, you find yourself easing away from the strict comparison mindset and into something calmer, almost aesthetic. You are no longer asking each particle to testify. You are just letting it exist under your lens. A cracked network looks like a drought map. A smooth surface looks like ice. A cluster of fine clay looks like fog. A mica plate looks like a shard of mirror. And you think, briefly, about scale again. The same physics that shapes a continent shapes a grain. Stress fractures, erosion, deposition, coating, cementation. Even the way clay coats a grain is like weathering varnish on rocks in a desert. Thin films, chemical residues, time made visible. So you keep scanning, not to solve anything, but to witness the patience of matter. And you notice a subtle thing that feels like a soft ending to a thought, without actually ending the night. The slide is not infinite. Eventually, you reach the edge of the mounted sample, where the grains thin out and the glass becomes clear. At the border, there is a strange quiet. Fewer particles, more empty light. It reminds you that every sample is partial. Every microscope field is a tiny window. You never see the whole landscape, only fragments. So when you make conclusions, you do it with that border in mind. You saw some of the truth, and you left plenty unseen. And with that quiet limitation acknowledged, you keep the microscope centered and let your attention drift toward the next practical step you have not done.